Okay, I'm going to start right into this. Before I do, I'd just like to share this disclaimer because this is an all-natural supplement. It is not intended to diagnose, treat, cure, prevent any illnesses or diseases, and the statements that I'm going to share with you have not been evaluated by the FDA. With that, let's talk about the human cell. Right here, we have a depiction of the cell, and the human body is made up of over 100 trillion cells. And in the middle, right here, is the nucleus. And I don't know if you can see them that well, but there's those little X-like things in there. Those are the chromosomes. So here's a chromosome blown up. And within the chromosome is something very important called the DNA. That's our genetic gene code, or the expression of the cell physically. So they sit in the middle of every cell, and they generate an expression which follows the cell, the physical cell follows that expression. Now, we've learned about telomeres, especially over the last decade or two. They're being represented by the red tips on the chromosomes here. And we now have learned and discovered that as cells divide and replicate new DNA, the ends of those chromosomes are wearing away, and those are called the telomeres. And that's not a good thing because that's like the cap that holds all of this together. So as it shortens, it's actually damaging the expression of the DNA. And they've now linked every symptom associated with aging and all the degenerative issues that we go through that affects the quality of our health. So as the telomere gets shorter and shorter, we experience more and more the severe issues associated with aging. Now that's called basal cell telomere shortening. That happens through normal biological processes, but it doesn't have to be that way. And I'll talk about that in just a minute. There is also, because of lifestyle, we've gotten to a place in our society where we're putting things into the body that are damaging it. Not only are we lacking the nutrition, but I'm talking now about things that are like poisons to the body, parabens, oxidative stress, and things that we're exposed to. That's called accelerated telomere shortening. So there's two types, the basal and the accelerated. So it's like we're shortening the candle on both ends, like a candle burning from both ends. We've sped up these symptoms that are associated with aging and disease. In fact, in April 2012, USA Today had on the front page that children that had been exposed to certain issues in their environment and in their life, their telomeres were shortened over and above basal by 10 years. So they had premature 10 years faster, which means they're going to develop these symptoms again associated with aging and disease, which that's not a good thing. Now, the resolve to this is innately within the chromosomes itself. It's something called a telomerase gene. And we're going to talk about Bill Andrews and his discovery and his role in helping us to understand more about what we can do in support of this telomerase gene. Now, to just get a little better visual, we're going to look into a skin cell here. So we're going to really magnify this. And within every cell, again, is that nucleus. So let's blow up the nucleus. And within the nucleus, we have the chromosomes and the DNA. Let's blow that chromosome up and identify with the yellow tips the telomere. When we're conceived, that telomere is 15,000 bases long. So let's stretch out that telomere and get a closer look at that blown up. So that's 15,000 bases long. When we're conceived and we're starting to develop in the womb, our cells are dividing, which means that telomere is getting shorter and shorter and shorter. And by the time we're born, we lost 5,000 bases. But our cells don't stop dividing as we continue to grow and develop and live throughout the length of our lifespan. Our cells continue to divide. So the telomere just gets shorter and shorter and shorter. When it gets to a certain point, it so negatively impacts the DNA that we start to now see these symptoms associated with aging. So again, when we're born, we have 10,000 bases left. And then as we continue to mature, those 10,000 bases get shorter and shorter, damaging the DNA more and more, until eventually that cell shuts down. And that's right about 5,000 bases. That's senescence within the cell. The cell shuts down and dies, and that's basically when we die, right around 5,000 bases. So born with 10,000, die right about 5,000. Bill uses the analogy of the shoestring and this plastic tip on the end of the shoestring. Pretend that's the telomere. When we hit right around 5,000 bases, it's like losing that plastic tip on the shoestring. It starts to fray and come apart. That's essentially what's happening to our DNA inside, so it no longer can give that expression. And so there's the process. That's a very simplified analogy. Now let's talk about the human telomerase gene, which Bill led a research team to discover in 1994. What's the role of the human telomerase gene? To stop that telomere from shortening, from preventing it from even happening in the first place. And he discovered it doing so in the reproductive cells, the actual sperm and egg cells. As we get older and older, the telomere and the gene expression stays the same in our reproductive cells. And so when Bill discovered that, he realized that was the resolve to these symptoms associated with aging, which he had taken on for his purpose in life to stop. And that was a big discovery. Up to that point, it hadn't been identified, and Bill was the one that was able to identify it and prove that. And then, knowing that that was the resolve to the symptoms associated with aging, he then tried to activate that gene so it worked more effectively in the rest of our cells. And by 1997, working with human skin cells, he was able to activate that gene in the human skin cells. That was a very profound thing to have take place. It was so profound that Harvard got involved, and a Dr. Ron DeFino got involved with Bill's methodology that he used to do that, and he implemented it in mice. And that's where everything really got exciting, and the research really took a big, like a turbo boost forward. There was less than 100 articles written on telomeres at this point, worldwide. So what did Harvard do with the mice? They activated this same gene in the mice, and they took these old mice, activated the gene, and all of the symptoms associated with aging were reversed. And it's a very detailed report, but they saw things like their hair grow back, their skin got soft and smooth, their activity level was restored to that of a young mouse. They started running and bounding around again. Even their mental or cognitive ability was restored. They had lost 25% of their mental capacity, and they activated this gene, and it was restored. Another significant thing was that their bone density was also restored as well. The glucose levels leveled out. Dr. Ronald DeFino said it was an amazing observation of what looked like the reversal of the aging process. So then Bill and his lab turns his attention to trying to find compounds that you and I could consume that would activate this gene in us as humans, because that's the ultimate goal, is to have it happen for us. And that's where there's been a huge obstacle. They tested over 304,000 compounds, and they did find some, but they were all too toxic for human consumption. And in 2009, I was in the audience that he was lecturing to, and he said, we called it the race to telomerase. Who's going to find compounds that humans could use to do this? Who's going to do it first? So I want to show you here, this is a graph that Bill had showed us in that lecture, and he said we tried over 304,000 different compounds, well over $30 million worth of testing. And they invented the equipment to do all of this, and in fact they have some of the only equipment in the world that allows you to test for telomerase and then to watch it. The length of the red lines is what we're looking at here. All of these were able to induce the telomerase gene in human skin cells in vitro, meaning in a petri dish. But these were all too toxic for human consumption. But he did get these human skin cells in vitro to act like the reproductive cells. So it was a huge understanding of what was possible with an inducer. Now, there's a collaboration that took place in 2009 between the geneticist Bill Andrews and world-renowned and master formulator John Anderson, who's formulated for over 600 companies during his career. He's always worked with natural ingredients, and he's formulated nearly 2,500 different nutritional products. And he's in working with all the companies that he did and all the experience that he gained and learned and traveling around the world and learning what other cultures were using holistically, he came up with a very proprietary and unique way of formulating with extractions and enzymatic processes. And that's why he was introduced to Bill Andrews, because they felt like they exhausted the natural world and they couldn't get the natural ingredients to work. And so they turned to synthetic chemicals and drugs. Well, here they are being reunited again, and I call it science meets wellness. Bill taking another effort at trying to get the natural world to be able to induce this telomerase gene with the help of John Anderson. So it's product B, it's called IsaGenesis, and it was released in August of 2011. And they continue to work together, Bill and John, in this collaboration, and they just released what we call a 4th generation product B. So I want to talk about the evolution of this product here. Now, Bill was, again, back on the stage and reminding us about the compounds that they had found and what they had done. And he said, we partnered up with John, but we really kind of underestimated John. What he did shocked us. We were not prepared to see what we did see as soon as he started sending formulations over to us. So John started working in his lab, putting together formulations based on what Bill had taught him about the telomeres and the telomerase, and then Bill would vet John's ingredients in the human skin cells in vitro. And he says, what I'm about to share with you, again, we completely underestimated John. And what he did with these natural ingredients, by my estimation as a scientist, I put him in the category of genius. In less than 100 attempts, remember, they tried over 304,000 and tens of thousands of natural ingredients. In less than 100 attempts, John was able to come up with 7 hits from all natural ingredients. I'm going to show you a really short clip here about with Bill sharing what they saw and what they felt about it in the first 2 hits that they found.
We'd seen for natural products, and we were pretty excited. It was the 11th compound we tested out of the first 15 samples that John Anderson sent us, and it was a hit. So we use the term hit in science a lot. So when we screen a lot of samples, we'll screen like thousands of samples at a time. But, you know, I have to confess, at first we thought, wow, was he extremely lucky? Because we thought, how could he only be asking us to screen 15 samples after screening thousands without a hit? We need a lot more samples than 15 if we're expecting to get a hit, but he got a hit. So he sends us another 15, and we get another hit.
That's pretty amazing. He actually left 2 voicemails on John's phone after finding those hits, saying how shocked they were and how excited they were over there, and would he hurry and call them back. They were very anxious to hear from him. When they released the product in August of 2011, they had found 12 hits. Well, 6 months later, they found 2 more because John's still working on this and wants to get it to the best that he possibly can. He's very ambitious. And so that became 2nd generation product B. And then what happened a year later is they had a special announcement. John, being ambitious, asked, what if we could really pass up these hits that you had over here that were unusable? And Bill told him, from natural ingredients, that would be impossible. You're not going to find that in a natural ingredient. So it says, loving a challenge, Anderson sent Dr. Andrews 80 to 160 different vials with formulations. 80 to 160. That's how ambitious John was. And then Bill's lab would report back every Friday. So then Anderson's hard work finally paid off with an exciting phone call from Dr. Andrews in early March. This would be 2012. You've exceeded our control. I can't believe it. What are you doing over there? And they announced the release of 3rd generation product B with these 2 hits that from natural ingredients that Bill told John would be impossible. It was at that time that Bill said from the stage, John's made me a believer. I now have this faith and belief in natural botanicals and John's ability to work with them to get to where we ultimately want to be. Well, John announced 6 months later that he found nutrients, like phytonutrients within plants, specifically in China, that had never even been found or known of. And he also announced that he found hits that were bigger than these. Well, they just released in August 2014, 4th generation product B, which has also gone through some clinical testing, which I want to share with you next. We are very excited about this. This is a game changer with this one right here. We're already seeing incredible results. From here to this, 3rd generation, was a big difference. We saw things with people's health faster. We saw things that we weren't seeing before. And this jump to 4th generation is even bigger. Let me share with you. There's an enzyme called catalase within the human body, and the body produces it naturally. And it helps to neutralize the free radicals, oxidative stress, the parabens and toxins that get into our bodies. And it's directly related through studies to the symptoms associated with aging. So the University of Arizona, the Nutrition and Health Promotion, did a clinical study. It's the gold standard clinical study that they did with product B. It's a randomized, placebo-controlled, double-blind clinical, meaning neither the doctors nor the participants know that they're taking the product. There's a sugar pill, and then there's also the product. So nobody knows about it, and they're just evaluating them based on the data. Well, what they understand from a study done at the University of Washington State, catalase has been shown to increase longevity in experimental research models by decreasing accelerated telomere shortening. So that's that other side of the candle, not the part that we just talked about with the hits and inducing telomerase in the normal human skin cells in vitro. The catalase helps to prevent that other side that's due to lifestyle. So here's the actual, some of the words from that study that was done by Washington State University. Mice engineered to produce high levels of the antioxidant catalase live significantly longer than their wild-type counterparts, by about 20%. In the article, they said equating that to humans, that would add about 24 years to the lifespan of a human. The results lend support to the free radical theory of aging, which attributes many of the infirmities of old age to accumulated cellular damage. They were able to identify that through this study and see that there was a direct correlation. Now remember, again, catalase decreases as we get older, and that's why we start to see this compounding effect of symptoms associated with aging. This validation has long been awaited. It's the most direct, testable proof of the free radical theory. So they were actually able to see it, document it, and write up all the papers on it. It's a good demonstration that fixing a cause can have an effect on a mammal. Now, we're mammals as well, and that seems like a no-brainer. Like, yeah, that's just obvious that if you were to fix the cause of the problem, you would see a direct result. But as scientists, it's very difficult to identify where the cause is. There's always a lot of theory around causes, but in this test, they literally were able to identify it, and it was a testable proof. So this is a big deal. Age-related problems including, and not limited to, DNA damage, cancer, and diseases like Alzheimer's. Due to a lack of catalase, or when catalase is in at a high level like it was with these mice, they didn't have these issues. Dr. David Sinclair from Harvard Medical School said it's a pretty big deal, and it is. So this is what was done. We tested catalase in this in vivo, in humans, this double-blind clinical study. Now, Suk Cho is the Chief Science Officer for IsaGenesis. And as he was telling us about this clinical from stage, he said, as researchers, we were so surprised by the findings that we double and triple checked the data, because it was results that they didn't expect at all. It was like out of the normal parameters of what you might think would happen. And he also said we were so excited as scientists because, see, they were able to identify the cause and see where it's at, what's causing it, be able to prove that, and then we're able to see a resolve to it through this clinical study. And it's not very often that a scientist gets to see the resolve to something that they know is taking place. It's something that's very rare where you can see a resolve to the issue that's been identified, especially in that particular scientist's lifetime. So here's what they found with product B's IsaGenesis in vivo clinical study. It showed an increase of over 30% in catalase production in humans, in vivo. That's incredible. That's a huge, huge margin of increase that directly affects that accelerated aging. And there you have it. That's an in vivo clinical study. We're very proud of that and very excited about that as well. It's a game changer for sure. 20% more active ingredients in this 4th generation, and it also has a new proprietary process of being more absorbable. So this is significantly more so than what was even done with the clinical study. This is 4th generation that we're talking about now, this IsaGenesis. It has been now proven to support liver, kidney, brain health, cell integrity, and healthy circulation. All of the opposite things that you would see with somebody who's experiencing those symptoms associated with aging. Again, this is a game changer. John Anderson is quoted as talking about 4th generation. He says, besides being the best telomere support product on the planet, that would be the basal telomere shortening, being able to support the telomeres so that there's not as big an effect on that basal cell through the cell replication. And then he says, IsaGenesis is also a very powerful antioxidant, which was shown through the clinical data. So there you go. We're helping to address both ends of that burning candle in a way that we never have before. So what we ask you to do is get back with the person that got you here, and they'll help you to get started on this product, 4th generation product B that's now available. You can order it now. And again, if you'd like to see the longer version that I go into more detail, just ask them where they can point you to where you could watch that. Thanks for joining me. Bye-bye.